We are here today at Select Breeder Services in Chesapeake City in the KWPN North America series of frozen semen for mare owners. I'm happy to come to Select Breeder Services in Chesapeake City and meet the founder and owner, Paul Loomis, to start our tour.
Welcome, Natalie, and I look forward to showing you our lab and what we do here at SBS. Instrument is called a flow cytometer, and it's something that we're now incorporating into our routine frozen blood semen analysis. This assay that we're going to look at now, you can do a number of different assays here. Basically, you stain the sperm with different probes that are fluorescent in different wavelengths of light. This machine has lasers in it that excite those fluorochromes on the sperm. It could be something that attaches to the acrosomal membrane of the sperm, or something that only penetrates sperm that have damaged membranes. There's a whole host of things we can do with a flow cytometer, including there is an assay for how intact the sperm chromatin is, or how intact the DNA is. Right now, this is some of the data that you see, and I'll just illustrate. This is a viability assay, and there's 4 quadrants here. This is the scatter plot, or the fluorescence for each of the objects that was in the sample. These are the sperm cells. This is debris. We can gate out just the sperm cells and look at the red and green fluorescence. And we plot the red versus green fluorescence here. Sperm that are fluorescing red have taken in propylium iodide. So they have damaged membranes, and it's an intercalating DNA stain. And what you see here, this is the quadrant of dead cells. You can see all of them up here. This is a plot of red fluorescence versus green fluorescence. This is the percentage of cells that are dead. And we know this because this is an experiment to validate this assay. This is a sample that we killed on purpose. So we flash froze it in liquid nitrogen, and when we thawed it out, and we flash froze it again, and we thawed it out. But this is what happens from damage associated with that. This is the quadrant of live cells down here, and you can see there's no live cells. This is almost all dead cells, and this quadrant up here is sperm that are sort of in transition between dead and live. There's a few cells in there, but not many. So this is our 100% killed sample. This is a normal sample that we haven't mixed any of the killed cells in with. What you see here is a population of dead cells, a population of live cells, and up here in this quadrant, a population that have taken up both stains. So they were alive and they're on the way to being dead. And then what we can do is mix in a known ratio the 100% live versus it's not 100% live because there's always going to be dead cells in any sample. But we mix these two together in known ratios to get different distributions. And then we can create a standard curve from that to validate the assay. But what it does is allow us, and you can see the different mixtures here and how they have resulted in different populations in these quadrants. What it does is allows us now, when we do a sample of fresh semen, a sample of cooled semen, or a post-thaw sample, to add to our motility data this viability information that tells us something additional to motility, what the difference between one stallion and the next, one sample and the next, maybe one treatment, one protocol, one extender type and the next, when we're trying to figure out what's the best way to freeze an individual stallion. You may have the same motility with 2 different extenders, and one may have a much higher viability than the other. That will give us additional information beyond what motility tells us, that allows us to make a better selection of what protocol to use for that horse. And this is the first assay that we're incorporating into our routine post-thaw analysis, but we will be adding to this assays of mitochondrial membrane potential. Mitochondria are housed in the midpiece of the sperm, and they're very sensitive to damage from cryo-injury during freezing. So we want a way to be able to measure how intact they are. Also acrosomal membrane integrity. because acrosomes can get damaged during freezing and thawing. Some stallions, even in their fresh semen, who have idiopathic or unknown reasons for infertility or subfertility, might have acrosomes that don't react appropriately to the stimulus in the mare's reproductive tract. So we can sort of simulate that in vitro and see if they react or don't react in an assay that we could do in the flow cytometer.
And now this particular machine, I mean, this is not something that's standard at all, even within university labs, correct? I mean, this is a very unique...
Well, it is a research tool more than a clinical tool. Prior to investing in this here in our lab, if we had a stallion with, let's say, for example, you have a stallion, you froze semen, his quality looked really good and everything, morphology was good, motility was good, but you were getting very, very poor fertility, and we might send samples off to a reference lab at a university where they could run some of these assays. But we're really excited to be able to bring this in-house and do it now ourselves.